हेलो फ्रेंड्स, so हम स्टार्ट करने जा रहे प्लांट किंगडम चैप्टर 3। In the previous chapter we looked at the broad classification of living organisms under the system proposed by Whitaker in 1969, wherein he suggested the five kingdom classification wise Monera, Protista, Fungi, Animalia and Plantae. In this chapter we will deal in detail with further classification within kingdom Plantae, particularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae through earlier classifications placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us also look at the classification within angiosperms to understand some of the concerns that influenced the classification systems. The earliest systems of classification used only gross superficial morphological characters such as habitat, habits, the color, number, and shapes of leaves, etc. They were based mainly on vegetative characters or on the androecium structure systems given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial systems gave equal weightage to the vegetative and sexual characteristics. This is not acceptable since we know that often the vegetative characters are more easily affected by environment. As against these natural classification systems developed, which were based on natural and affinities among the organisms and consider not only the external features but also internal features like ultrastructure and anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidences. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all characters, and the data are then processed. In this way, each character is given equal importance, and at the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that uses the chemical constituents of the plant to resolve confusions, are also used by taxonomists these days. Algae, algae bearing simple thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi, lichen, and animals, for example, on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and filamentous forms like Olothrix and Spirogyra, which is located in Figure 3.1. A few of the marine forms, such as capes, form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into the thallus. Asexual reproduction is by the reproduction of different types of spores, the most common being the zoospores. They are flagellated, motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Olothrix, or non-flagellated, non-motile, but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Odoterina is termed as angiospermous, anisogamous. A fusion between one large non-motile static female gamete and a smaller motile male gamete is termed oogamous. For example, on wax papers. Figure 3.1 algae. Green algae, Volvox, Olothrix, dictata. Green algae, Volvox, Olothrix, dictata. B. Brown algae, Laminaria fucus dictata. Red algae, Porphyra polysiphonia. Algae are useful to man in a variety of ways. At least half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy rich compounds which form the basis of the food cycles of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae of hydrocolloids, water holding substances. For example, algae, brown algae, and carrageen, red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria are used to unicellular algae rich in proteins is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyaceae, Phaeophyaceae, and Rhodophyaceae. Chlorophyaceae. The members of Chlorophyaceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplasts may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of the members have one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein. Besides starch, some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made on of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The asexual reproduction shows considerable variation in the type and formation of sex cells, and may be isogamous, anisogamous, and oogamous. Some commonly found green algae are Chlamydomonas, Polvox, Ulothrix, Spirogyra, and Chara. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous forms, ectocarpus, ectocarpus to profusely branched forms are represented by capes which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of the xanthophyll pigment fucoxanthin present in them. Food is stored as complex carbohydrates, which may be in the form of laminaria and mannitol. The vegetative cells have a cellulosic wall, usually covered on the outside of by the gelatinous coating of algae. The protoplast contains the addition in addition to plastids, centrally located vacuole and nucleus, the plant body is usually attached to the substratum by a hold fast and has a stalk. The stipe and a leaf-like photosynthetic organ and the front. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by flagellate zoospores that are pear shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the Ogonium oogamous species. The gametes are pyriform, pear-shaped, or bear two laterally attached flagella. The common forms are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus, which is located in figure 3.1b. Rhodophyceae. The members of Rhodophyceae are commonly called as red algae because of the predominance of the red pigment, the rhodophycoerythrin in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well lightened regions close to the surface of water and also at great depths in occurs where relatively little light penetrates. The red thallium of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as for Floridian starch, which is very similar to amylopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile spores. 3.1 divisions, figure 3.1 table, 3.1 divisions of algae and their main characteristics. Common name: green algae, chlorophyllaceae. Chlorophyll and B are major pigments. Stored food is starch, cell wall is cellulose, the flagellar number and position to 8 equal, apical habitat, fresh water, brackish water, salt water. Phaeophyceae, brown algae, chlorophyll A, C, fucoxanthin, mannitol, laminar in stored food, cell wall, cellulose and algin. Flagellar number 2, unequal lateral habitat, fresh water, brackish water, salt water. Red algae, rhodophyceae, chlorophyll A and D, phycoerythrin, Floridian starch, which is stored food, cell wall, cellulose, pectin, and polysulfate esters. Flagellar absent, habitat fresh water, some brackish water, salt water, moist. Gametes, sexual reproduction is oogamous and accompanied by complex post fertilization developments. And the common members are Polysiphonia porifera, which is located in figure 3.1c. Gracilaria gelidium. Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. First, liverworts. Bryophytes. First, liverworts. Marchantia, female thallus, male thallus. A is female thallus, B is male thallus. Mosses. Funaria, gametophyte, and sporophyte. D is sphagnum gametophyte. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession or bare rock soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus like the prostate or erect and attached to the substratum, while unicellular or multicellular rhizoids. They lack true roots, stems, and leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate antherozoids. The female sex organ called orchigonium is flask-shaped and produces single egg. The antherozoids are released into water where they come in contact with the archegonium, and antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called the sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but more but some mosses produce food for herbaceous mammals, birds, and other animals. Species or sphagnum moss provide peat that have long been used as of their capacity to hold water. Mosses along with lichens are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid, for example, Marcantia. The thallus is the plant one. The thallus is the dorsiventral and closely pressed to the substrate. The leafy members have tiny leaf-like appendages and two growths on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structures called gamma. Syngama. Gamma are green multicellular asexual buds which develop in small receptacles called gamma cups located on the thalli. The gamma become detached from the parent body of germinate to form new individuals during sexual reproduction. Male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into a food seta and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes, mosses. The predominant stage of the life cycle of a mosses is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is creeping, green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction is mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archiconia are produced at the apex of the leaf face shoots. After fertilization, the zygote develops into the sporophyte consisting of a foot seta and capsule. The sporophyte in mosses is more calibrate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum, which is located in Figure 3.2. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals evolutionarily. They are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places through some may flourish well in sandy soil conditions. You may recall that in bryophytes and the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, and the main plant body is the saprophyte, which is differentiated into two root, stem, and leaves. Figure 3.3. These organs possess well differentiated vascular tissues. The leaves in pteridophyte are small microphytes, as in Selaginella, and large macrophytes, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophytes. In some cases, sporophytes may form distinct compact sporangia, produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free living pteridophytes. फर्स्ट फिगर है हमारी सिलेजिनेला की, सेकंड है इक्विसेटम, थर्ड है फॉर्न्स एंड फोर्थ है साल्विनिया. सिलेजिनेला, लीव्स, स्टेम्स, रूट्स. इक्विसेटम की है हमारी सेकंड, थर्ड है हमारी फॉर्न्स की, और फोर्थ है हमारी साल्विनिया की फिगर. Mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of antherozoids. The male gametes released from the antheridia to the mouth of archegonium. Fusion of male gametes with the egg present in the archegonium result in the formation of zygote. Zygote is thereafter produces a multicellular, well differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called two kinds of spores. Macro, large, micro, small spores are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophytes in these plants are really retained on the parent sporophytes of variable periods. The development of the zygotes into young embryos takes place within the female gametophytes. This event is a precursor to the seed habit considered as an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Xylotum, Lycopsida, Selaginella, Lycopodium, Sphenopsida equisetum, and Pteropsida Dryopteris adiantum. Gymnosperms. The gymnosperm seeds are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. Gymnosperms include medium-sized trees or tall trees and shrubs. For figure 3.4, one of the gymnosperms, the giant redwood tree sequoia is one of the tallest tree species. The roots are generally tap roots. Roots in some genera have fungal association in the forms of mycorrhiza pinus, while in some others, cycas, small specialized roots called coralloid roots are associated with nitrogen fixing cyanobacteria, may be simple or compound. Nitrogen fixing may be simple or compound. The cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are born on sporophylls, which are arranged spirally along an axis on sporophylls which are arranged along an axis to form lax or compact strobilia or cones. The strobilia bearing microsporophylls and microsporangia and are called microsporangiate or male strobilia. The microspores develop into a male gametophytic generation which is highly reduced is confined to only a limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophys with ovules or megasporangia are called macrosporangiate or female strobilia. The male or female cones or strobilia may be born on the same tree, Pinus. However, in cycles, male cones and sporangio megasporophylls are born on the megaspore mother cells is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovule are born on megasporophylls while may be clustered to form the female cones. The megaspores, one of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears two or more archegonia or female sex organs. The multicellular female gametophyte and is also retained with megasporangium, unlike bryophytes or pteridophytes. In the gymnosperms, the male and female gametophytes do not have an independent free living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules born on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their currents near discharge their contents near the mouth of the archegonia. The following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered. Gymnosperms, Cycus, Pinus, जिंकगो साइकस फर्स्ट है हमारा साइकस, सेकंड है हमारा पाइनस, थर्ड है हमारा जिंको. Angiosperms. Unlike the gymnosperms, where the ovules are naked, in the angiosperms and flowering plants, the pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest wolfia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes: the dicotyledons and monocotyledons. Angiosperms, dicotyledon, monocotyledon. समरी, तो यहाँ पर हम समरी स्टार्ट करते हैं. Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Algae are chlorophyll bearing simple thalloid, autotrophic, and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three types, namely chlorophyllaceae, phaeophyllaceae, and rhodophyllaceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes, which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction, and their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum of rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axes bearing spirally arranged leaves. The main plant body of a bryophyte is a gamete, producing, and is called a gametophyte. It bears the male sex organs called antheridia or female sex organs called archegonia. The male and female gametes fuse to form zygote, which produces a multicellular body called a sporophyte, and the produces haploid spores. The spores germinate to form gametophyte. The pteridophytes, the main plant is sporophyte, which is differentiated into true roots, stems, and leaves. These organs possess well differentiated vascular tissues, and sporophytes bear sporangia, which produce spores. The spores germinate to form the gametophytes, which require cool, damp places to grow. The gametophyte bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of male gametes to archegonium, where zygote is formed. After fertilization, the zygote produces a sporophyte. The gymnosperms are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed, and therefore these plants are called naked seeded plants. The gymnosperms produce microspores and megaspores, which are produced in microsporangia and megasporangia, born on the sporophylls. The sporophylls, microsporophylls, and megasporophylls They are arranged spirally on axis, and pollen tube releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. While following fertilization, the zygote develops into embryo and the ovules into seeds. The angiosperms are divided into two classes, the dicotyledonous and monocotyledonous. तो यहाँ पर हमारा द प्लांट ख़त्म होता है, जिसमें हमने पहले से देखा अलगी, उसके बाद हमने ग्रीन अलगी, ब्राउन अलगी एंड रेड अलगी देखा, उसके बाद क्लोरोफाइसी, फियोफाइसी, रोडोफाइसी, ब्रायोफाइट्स, लिवरवोट्स, मॉसेस, टेरिडोफाइट्स, जिम्नोस्पर्म्स, एंजियोस्पर्म्स। तो उसके बाद हमने समरी देखी। तो यहाँ पर हमारा द प्लांट किंगडम ख़त्म होता है। सो थैंक यू सो मच फॉर वॉचिंग।